everyone's presentations today, as well as the other symposiums, and I'm looking forward to present my research with you. Also, I'd like to let you know that I'm going to be using QR codes throughout my slides, so feel free to take pictures as you see fit. So, as life continues to evolve throughout all domains of life, one critical component is shaping natural selection, and thus the composition of genomes, is environment. Environments impose selective pressures in the form of climate, localized temperatures, oxygen content, as well as pressure, to name a few. These selective pressures impose impacts on the evolution of genome composition, impacts that are currently under-examined. When we look at extreme environments, including hydrothermal vents, salt lakes, hot springs, as well as volcanoes, these environments impose incredibly selective pressures that make it very difficult for life to sustain. Despite being highly selective, organisms across all domains of life have adapted to these extreme environments and are aptly called extremophiles. It's also been observed that within these extremophilic categories, there have been phenotypic adaptations that have converged to permit this adaptation to extreme environment, such as the evolution of antifreeze proteins found across all domains of life. At the bottom here, you can see a chart that categorizes the different temperature and pH-related categories of extremophiles. These extreme phenotypic convergence in extremophiles have been found to be underlain by genomic codon usage bias, such as the adapted use of increased or decreased codons throughout psychrophiles, which are cold adapted. These unique codon patterns are also associated with different extremophile categories, like the thermophiles, which are adapted to extreme temperature, acidophiles adapted to extreme acid, and alkalophiles, which are adapted to extreme alkali. The ability to assess these codon usage patterns have been permitted through the advances in next-generation sequencing. Another observation that has been found through next-generation sequencing is the founding of the genomic signature. So the genomic signature essentially describes global, pervasive patterns of oligonucleotide compositions found in a species-specific manner. These oligonucleotides can range in size, with the also known as k-mers, with the size of the oligonucleotide described by the size of K. And they can be used to facilitate highly accurate taxonomic classifications due to their species specificity. At the bottom here, you can actually see the molecular distance map that highlights how distinct genomic signature features can delineate species at a high accuracy at the kingdom level. Since genomic signatures are found prominently across the entire genome, you can take the entire genome or even a segment and visualize this genomic signature or oligonucleotide patterns with a CGR or chaos gate representation. This is a technique that takes a whole string of nucleotides like a genome assembly or a fragment and plots them in a fractal-like arrangement. It does so by plotting each consecutive nucleotide in a k-mer a half distance to its respective corner. For example, the trinucleotide ATG would be represented in the space here highlighted in red, where a dot would be made to symbolize its presence in the genomic signature. You can see in the middle row here how these CGRs are extremely specific to the species, and they can be used to classify bacteria and archaea from each other. Seeing these very distinct genomic signature features between the bacterial species on the left and the archaea on the right. However, while recognizing that these are incredibly impactful, they're very, very detailed and difficult for the human mind to kind of break down. So we can visualize them and resolve them through a FCGR. The FCGR essentially overlays a grid on top of the CGR and allows for the k-mer counts to be counted with more kind of understandable grid spaces. So you can kind of resolve the signature down to a FCGR, which is more comparable to a QR code. We actually investigated the CGRs and the kind of genomic signatures associated with temperatures as well as extreme pHs in a paper that we recently published, as well as discussed in a poster last year at the 2023 EMGS presentations. We found that there are distinct environmental signatures associated with extremophiles adapted to extreme pH and extreme temperature, and you can see the distinct genomic signature features in the FCGRs of their adapted species. However, extreme pH and extreme temperature are just the tip of the iceberg. Radiation is a well-known mutagen mutagen, genotoxin, and environmental hazard. For example, the high ionizing radiation or high energy ionizing radiation of gamma rays and X-rays has been shown to induce mutational impacts in the form of DNA protein crosslinks, base damage, single-stranded and double-stranded breaks, as well as bulky lesions. Additionally, lower energy radiation, such as ultraviolet and infrared radiation, has also been shown to induce DNA damage. However, this being said, many organisms are adapted to this extreme radiation, like Deinococcus radiodermis, a well-characterized radiotolerant organism that can survive in the high-energy gamma ray environment of space up to a whole year. This is a bacteria, and it has enhanced DNA repair, and that enables it to survive in these extreme environments. A eukaryotic example is Recursius coronifer, which is a tardigrade or water rat, more commonly known, that's also capable of surviving in space and has been able to live there for a whole month. Therefore, since other extreme environmental factors like pH and temperature have been shown to impose distinct impacts on the genomic signature composition, we ask whether radiation imposes such an effect. We investigated our hypothesis using machine learning tools as well as a novel data set for microbial genomic signature analysis. This data set was essentially curated through literature search techniques that identified radiotolerant microbes, as well as integrating with the DET-R database, which was built back in 2021 that aimed to collect prokaryotic radiotolerant organisms, as well as their genomic signature features, genomic assembly features, and metadata. We also wanted an outgroup, so to speak, so we collected a kind of one-to-one ratio of non-radiotolerant organisms to radiotolerants. So we did this through finding related bacteria or archaea that were found from the same family or order that were not adapted to extreme environments or extreme radiation in particular. For the machine learning model, we used 6 different classifiers integrated with 10-fold cross-validation. So this cross-validation essentially breaks up the data set into 10 bits and uses each bit as a training or testing data set. So it repeats this iterative training and testing process until essentially an average signature and features are built and learned by the model. We also tested the 6 different classifiers as well as different fragment lengths and analyzing at different k-mer sizes to kind of find that sweet spot of what is the best criteria for classification of these genome assemblies. There, it was found that the MLDSP1 algorithm, which utilizes the linear discriminant classifier, the fragment length of 100 kilobase pairs, and the reading frame of k equals 6, or hexanucleotide, resulted in the best classification accuracy for environmental classification. We found 88.5% accuracy for this classification of radiotolerant versus non-radiotolerant, as well as quite high, medium to high accuracies for other parameters tested. We also tested the classification for taxonomic levels across phylum all the way down to genus, and we found extremely high to nearly perfect classification accuracies, which demonstrates the robustness and the capability of the model to classify both in the taxonomic and environmental context. Now, it's important to address some of the limitations. So, while it's really great that we're able to classify organisms if they're adapted to radiotolerance or non-radiotolerance, we don't know the metadata associated with the dose level, the duration level, and the radiation type. One would assume that the radiation exposure of an X-ray environment, which is much more higher energy ionizing radiation, would have a more significant impact than lower energy UV radiation. So being able to annotate our data set in terms of the dose level, the duration level, and the radiation type would be really great, because then we'd be able to kind of get a measure of how these different radiations and different exposure types can affect the genomic signature composition. Another level that we would be really interested in adding is the other metadata associated with the kind of growth and habitat of these species that we investigate. So that includes the salinity growth ranges, the pH and temperature, to kind of encapsulate the complexities of polyextreme felicity. Overall, it's really great that we demonstrated this kind of genomic layer of radiotolerance. Previously in the data, or in the literature, the kind of mechanisms that underlay radiotolerance are more biochemical and associated with antioxidant defense, cellular cleaning, and DNA repair. But through our investigation, we've actually shown that there's a genomic compositional level associated with this radiotolerance. So this is really great for not only environmental taxonomy or environmental classification, but also understanding how radiotolerant organisms can adapt and live in these extreme energy mutagenic environments. I'd like to thank my supervisor, Dr. Kathleen Hill, as well as collaborators Manire Safari, Dr. Leela Kari, and Dr. Rajiv Randhawa, as well as our funding resources, NSERC, the Western Research, and Compute Canada. Here are all the QR codes used throughout my presentation, so feel free to take a photo if you'd like, and be happy to answer any questions.